Hello, everybody. My name is David Leclerc. I'm the host of the ChUG podcast. I am extremely pleased today to host Oliver Burton. He's assistant research professor in Cambridge. How do you do, sir?
Yeah, doing really well. Thanks for having me on.
Mine, quite a pleasure. Uh, it's our first time speaking together, and I got to say, you're on my mind quite often these days. I've been helping my users with their unmixing issues over the last few weeks. Like there's a quite a high number of people that are having trouble, and whenever I help people, my mind go into this Rolodex of your blog, cytometry.com, and I try to find the post that best describe the problem we're trying to fix. Uh,
Thanks.
Whenever people come to me and say, "Oh, I've done the overnight staining protocol," I just know that it's the overnight staining protocol that you've described. For some reason, somebody brought a tube of
Oh, fancy
dishwasher
that.
liquid, and
Yeah.
so this is my one of my users thinks I'm going to do a I'm going to test your protocol for using the fixation with the dishwasher liquid. So it's more work for me. What's up with that?
Well, I mean, I should point out I am certain I am not the first person to do overnight staining. I've just done some work on it to make it more accessible to people. Like I'm not gonna like say I came up and invented that. Like we put it out there as like a way to do cytometry that makes it cheaper for people.
Is
But
that
I suspect people have done that before. I think if you look in papers, you'll find that people did overnight staining before. Like you do overnight staining for ELISAs and things, right? And for Westerns. It's
right?
not like
Is the idea of using dishwasher soap a novel one, or like you basically the reagents are known, and so it's kind of like it came to mind to everybody, but like you're the one who just pushed the idea to.
I think that's new.
Mhm.
It's something I've been trying to solve for years because, I've done a lot of work with transgenic mouse lines and particularly in the field of regulatory T cells, where you always need to stain for your FOXP3 to know that you're working with your Tregs and that's the nucleus, and then you're trying to do these fancy reporters to track like different cytokines to know whether they're still Tregs or, tracking fate mappers. And it's just really hard with all the existing protocols to get good representation of your transcription factors with cytoplasm or your GFP or your cytokines. And that's been a struggle since I was a PhD student. And so when I got hired by Adrian Liston several years ago now to be a flow guy in the lab, that was one of the things I set out to work on was, well, can we actually fix this problem a bit? Because they were doing a ton of this work of, uh, working on extensively on Tregs and trying to do fate mapping with RFP and GFP and YFP and look at cytokines. And we needed something better. And so I looked at a lot of the stuff that was out there. There was some protocols out there where people had done good work on it, but it wasn't really quite there. And there was some out there stuff too. Like one of the really cool things is that you can actually put the cells in a microwave.
Right.
You just chuck your cells in the microwave for like 30 seconds, and then you can stain the nucleus. You lose like 99% of the cells, but the ones that come out, you can actually stain.
Right. I think I was aware of that protocol. But like just heating food in my like depending on the different
Yeah,
microwave you're going to use, you're going to get
exactly.
completely different results. So I
Absolutely. That's always occurred to me immediately. It's like, this is not going to be reproducible,
right.
but it's fun.
Yeah. Hey, listen, my roadmap is just basically hitting different topics that you covered in your blog. But I think we should probably start by talking a bit about your career and where you're at right now. Can you like I just know you through your blog, and so can
Hmm.
you basically flesh out how you got where you are right now?
Sure. Um, so let's see. I started out my PhD in Cambridge here, where I am now. I'm an American though, so I came over here to study because I wanted to see some of the world. I did immunology in a lab led by Professor Ann Cook, who's now retired, and that was working on autoimmunity and innate immunity and trying to understand like how we can modify the immune system such that we can redirect it away from an autoimmune response to more to a tolerogenic response. And so that was my main interest at the time, was trying to understand how we retrain the immune system so that it's not damaging the self. But basically I fell in love with cytometry doing that because we were doing a lot of flow, and I thought that the coolest room was the cytometry room because we had this technician running the core, Nigel Miller, and he had one of these MoFlows.
Mm.
I think it's the MoFlow Legacy. I'm not certain on the name, but it's the one that it sits on a floating table, and you have these giant black box lasers, and you manually align them by shining a mirror on the ceiling, and there's oscilloscopes and giant banks of switches. And I thought, oh, this is really cool. I don't understand how most of this works, but seems like an awesome thing, and all I have to do is bring in my cells, and I get this cool data out of this fancy machine. And then, so I spent a lot of time just learning more about cytometry during my PhD and really kind of learning more about what you can do with it and how we can basically adapt all of these different assays like proliferation, immune response, cytokine secretion to cytometry. And so that just became my go-to tool. Rather than doing an ELISA, I would do cytokine staining. I then moved to doing a postdoc in Boston, working on food allergy, so sort of a different way of where the immune response can go wrong. And I started going down the track of becoming like a group leader, trying to get funding and become independent, working under my mentor then. And that quickly became quite stressful for me. And I realized I didn't really want to do that if I could find a way out of it, because it seemed quite similar to what my father had done as like an independent contracting self-employed businessman, where you're constantly worried about providing funding for the people under you, like keeping those people employed. And it was too stressful for me to go down that route. And so that, combined with getting back together with my current partner now, meant that I moved to Europe. So I saw this job opening in Adrian Liston's lab for a flow cytometry specialist in Belgium, and thought, what the heck, I'll apply for that. I'm pretty sure I sent in my application after the deadline. So I got a very nice rejection letter from the secretary immediately.
Right.
But anyway, ended up getting the job and spent the first year there basically really focusing on getting better at flow cytometry and developing these high parameter panels that would be robust. And that was really my intro to thinking about it in a rigorous way rather than just doing it and trying lots of stuff to do this. Like that was more like, all right, let's do some systematic testing of what works
Mm-hmm.
and what doesn't work. And I knew sort of like, well, these are the problems. Like how can we go out now fixing them? And so since then, that's been a large part of what I do in the lab, is like thinking, all right, these are the problems that we're facing either in our cytometry work or in other aspects of our immunology work. What can I do as like the senior person who has more technical experience to make that work more reliably and better for people in the lab so that we get sort of better, more reproducible results, and it's just easier for everybody.
Yeah, there's a lot to unpack here. I'll point out that you mentioned the MoFlow legacy, and I have this idea that the legacy is similar to listening to Led Zeppelin and getting hooked on great music, or starting drinking German beer, and that's what stops you from drinking Miller Lite, so you can't go back anymore. Like this is the machine that gets people excited about the technology, like overall, like anybody who touched the machine gets like hooked to flow cytometry. It's kind of cool.
Glad to hear I'm not the only one.
Oh yeah. I was trained on a MoFlow. It was just such an awesome machine. The the idea. So whenever I think of you, and there's people like you in my research community where they basically outclass me in terms of how to stain and how to use the instrument after a while, because they use the instrument constantly, and I can train the basic stuff to people, but like they get so proficient with it that like
Yeah.
I start talking to them and they become and so that's the what's your relationship at some point with I assume you have a core facility that you work with, or
Yeah.
how does the relationship builds up over time with these people? You keep touch, or you at some point do you feel like you're just completely um, self-sustained in this environment, and you maybe do not need their support as much?
No, I mean, I love their support. It's
Yeah.
great. I really rely on them. Like there's a whole bunch of stuff about like the maintenance of the machine and the way the innards work that I don't understand, and I don't have to because they take care of all of that. Maybe I should, and I'd be a better cytometrist if I understood like how a PMT really works. But there's a lot of other stuff that I can focus on and get good at because I have their expertise,
Right.
right? And that's kind of the nice thing about science in general is we distribute the expertise so that you can specialize and then advance.
Yeah.
Um, yeah.
And the other thing, so start getting back to your blog, and in the position that you're into, where you basically are trying to resolve issues for everybody and make everything standardized and everything. Like if people have an idea of what they want to do, they'll have a roadmap to that you kind of provided for them. Um, Writing these things in the blog, where did what drove you to like start the blog to begin with?
Yeah, I mean, so I think that part of it is that, I'm quite privileged to want to be able to be a scientist. Like the background that's allowed me to enter this pathway and achieve this is because I've had a lot of advantages in life. And then I've had the advantage specifically in my career of being given this sort of funded, protected position where I don't have to focus on the usual parts of being a senior scientist, of getting funding. Um, and I can focus on the tech part. And that means that I have done a lot of stuff where I see it as like I've made the mistakes. And now if I share that information, maybe somebody else doesn't have to make the same mistake. Um, and so there's a whole lot of that because I do a lot of stuff and I make a lot of mistakes. And I learn from that. But a lot of that doesn't get published, right? Because it doesn't fit into your scientific papers. Some of it fits into technical papers, things like OMIPs, but even there, like most of it's about like this particular assay using a lot of information that we already have. And so there's just so much of it that would get lost otherwise. And I'm sure that other people are doing this as well, right? And it's also getting lost there. Like, so everybody's repeating the same optimization steps. And so a large part of it was, well, I want to not have everybody waste time, energy, resources repeating the same things, repeating the same mistakes to achieve the same result. And it's a lot of work to publish this, and I don't need to publish this for my career. Let's just put it out there.
Right.
Um, so that was a large part of the motivation. I was also, at the time I started it, a bit burnt out, and looking for something to do that was more interesting and considering leaving science. So I thought, well, I have a duty to write this down
Mhm.
and share it with people so that it doesn't get lost.
Wow, that's fantastic. It's like from like that place to get a tool that's you. I don't know if you realize how often your blog posts get cited in the Discord channel, for example, in Cytometry. Regularly somebody is going to start
Sure.
listing all the posts that are relevant to the discussion, and just like
Okay.
it's kind of, I don't know, you're
Cool.
having a huge impact here.
Well, that's good. I mean, yeah, I don't know. I'm trying to strike a balance with being clear about like what I don't know on there, what
Mhm.
is useful, and like that it's not like I'm trying to be the first person to say that this is the way, this is just my experience. But if that's useful, that's great because that's what I want it to be. I want it to be helpful for people.
Yeah. The I Because you mentioned like publishing in your failed results in papers and stuff, and the nice thing about blogging is also that you develop a storytelling uh, aspect to it. Like I find most papers unreadable just because the structure is so
So
rigid,
fixed,
and you
right?
can't really visualize the story of what's going on in the paper. I stopped my training at the masters and I was done after that. So I don't have
Yeah.
the drive of the PhD people to read papers and understand everything in it. But like
I
give me a
don't read
good
that
story in the
much.
blog and like this fits in my like it basically the information that's in the blog fit better in my mind afterwards. It's basically here.
That's nice. It's also like usually I can focus on like a particular nugget that might
Mhm.
be like one figure in a paper or two and really kind of break it down. Um, and you can show bad data
Right.
because the point is, well, this is the problem.
Yeah. Yeah.
And I'm have to say also like I'm senior enough and I'm not at risk of losing my job and I can put out bad data and it's not going to make me
Right. Right.
suffer in a career, which is nice also.
Yeah. All right. So if we were to go to some of the major items on your blog, some of the bigger ideas, we already discussed the fixation with dishwasher soap. Um, maybe just to put a final touch on it, like so essentially we're using dishwasher soap as a replacement for super expensive kits that are available commercially that will work uh, most of the time for some projects or basically but like using this technique basically reduce the cost and um, I saw one of your latest post is basically the caveats of using
Yeah,
so do you want to give a few words about uh, basically
sure.
what would the general idea like let's put it like how do you fix and then where does it get crunchy.
Yeah. So I mean the general problem is that we're trying to access different parts of the cell at once. And that's kind of difficult because each of those has different sort of biophysical chemical properties. And we're trying to get these big antibody fluorophore into these different cellular compartments, which means making big holes in the membrane. And when we do that, things can leak out of the cell. So depending on what chemicals you use to stabilize the cell, and then what other chemicals you use to punch holes in the membranes, you can get those big macromolecule antibody fluorophore into different parts. And you can also cause more or less things to be lost in terms of falling out of the cell or being cross-linked in such a way that they no longer are reacting to the antibody. And so there are kits that are specialized to do sort of one thing or another, like for cytokines or for transcription factor staining, and they're really focused on getting us access to that compartment of the cell. And that is great. Those kits work fantastic for what they're supposed to do. But the problem is that oftentimes we want to look at different parts of the cell at once. And that's what I've been trying to figure out a way of doing that. And this is the best solution that I've been able to come up with over the course of several years of working on it off and on to get something that allows you to do cytokines and transcription factors at the same time, or transcription factors and, say, your GFP reporter at the same time in a way that's both accurate and sensitive. It's also cheap. To be honest, that's not usually the big issue because usually your antibody is the expensive reagent.
Right.
But it's nice that it's cheap. It's not going to be as good as a kit that's dedicated to getting due to the nucleus for nuclear staining. It's not going to be probably as good as a kit that's dedicated to cytokines for doing cytokines, although it's pretty close in that case. But what it does allow you to do is put these things together, which is what we need a lot of in complex mouse experiments, but also increasingly now for things like CAR T cell work, where you're transducing your CAR T cell with your CAR, but also maybe a GFP reporter to track whether they're transduced, and that's how you're measuring whether which ones are transduced, and then you want to look at like what transcription factors they're expressing to know if they're stem-like or whatever. So being able to put those together was really the goal of the protocol. So yeah, there's a bunch of caveats where, if you're trying to do one thing specifically, yeah, just use the kit that's for that. But this is a general purpose protocol that works pretty well for a lot of stuff.
Cool. Um, Another one that I mentioned is the overnight staining, and I remember a paper coming out that came out a few years ago, and that made a big splash. It was basically discussed quite a bit, and so here the idea is basically you fix your cells and you just stain overnight. Uh, I can't remember the details. I this is again, podcasting is not my job, so I don't prepare as well as I should
Yeah.
for this discussion. But I don't remember the details of the protocol and whether or not at that point titration becomes more or less important, or since the staining time is so long, you can start with a reasonable amount of antibody and then get decent results at the end. Can you this like basically describe what your how do you see this protocol and how it fits in?
Yeah, I mean, so I think that you're right that you're not very clear on the details because I probably wasn't very clear on the details
Oh,
in the paper because
put
to
that on
me it's
yourself.
like,
This is me. This is
well,
me.
no, it's an approach, right?
Yeah.
So it's you can change the fixative. You can fix or not, although I always recommend doing the fixation. But depending on what approach you use, it's sort of a different protocol. So it's more like the longer you leave it, the better sensitivity you're going to get generally. Um, Not with every antibody. Not everything works after fixation. Um, you need to test that. And in
Meaning
some
at some point,
cases,
like the antibody, like the time that the antibody is just no good after fixation for that specific
yeah,
epitope.
sometimes the epitope's just completely cross-linked or ruined by the fixative, right?
Yeah.
Other times the antibody is just too non-specific once you expose it to the inside the cell because there's just so many more proteins for it to bind to. Uh, so a lot of the chemokine receptors, for instance, are, at least in human, are hard to stain after fixation. It's just not very specific. You can get signal, but it's not very clean. Um, so I don't know if the titration is more or less important. I suppose it's probably slightly more. A lot of the time, you have a much wider range of possible values with the titration after fixation. So maybe your surface staining, you're usually looking at somewhere between 1 in 20 and 1 in 200 as your dilution factor. Whereas with the overnight, we have some that go down to 1 in 10,000. And I know that other people have pushed that a lot further in some cases. I generally don't because if it goes down that far, I find that, well, you're going to be pipetting amounts that are unreliable. So don't do that. Um, yeah, so I mean, it's more about just that the longer you leave it, if it's something that you're struggling to get staining on because of steric hindrance or low expression levels, usually you can get better signal to noise ratio by leaving it for longer. And it doesn't have to be overnight. Like a lot of those chemokine receptors on the surface, you can leave them for 2 hours, 4 hours, and you'll actually get a nicer separation than if you do the standard sort of 30-minute separation staining.
Is there such a time as over like a too long a time of staining, or at some point it's just like you get to, it kind of max out and you're good?
So apparently not.
Okay, that's good.
I don't know how long.
Like until the cell starts breaking apart due to
Yeah, but if they're fixed,
yeah, it's gonna be a
right? So I
very very long
mean,
time. Yeah.
I know that I've left fixed cells in the fridge after staining and couldn't see a difference running them a week later.
Oh,
So I also know that one time I did the overnight staining over the weekend and it was fine.
right.
So it seems to sort of stabilize after about 16 hours. I really don't recommend doing longer than one
Yeah.
night, but
Well, when the
also if you forgot
fluorophore
about
at
it,
some
just
point
don't throw
start
them
breaking
away.
apart,
Sorry?
would the fluorophore at some point start breaking apart? Because I know for some other applications, so Luminex
Yeah.
multiplexing, uh, we're told after 3 days your PE starts breaking apart and the signal goes down and less reliable results at the end. So I'm not sure if that's
Yeah, I
that
guess. I mean, it depends on what they're in, right? Because
right.
Is the PE breaking apart in the antibody vial? Probably not so
No,
much.
after so you stain the plate, the beads are stains, and then you've run them, for example, and then you wait for the users to tell you
Yeah.
what to do with the plate. Do you need to run again because some data looks weird? And then the rule of thumb, the lab from tech support is like after 3 days, like forget about it. It's not as
Yeah, I mean, I guess the question is then what's causing the breakdown because
right.
it's not happening in the antibody vial before you put it on the beads or whatever, right?
Yeah,
So,
yeah,
but yes, you need to be careful about that sort of stuff.
right. What I like about this overnight staining protocol is also that it it's a nicer workflow generally for balance of life and work, and it also allows you to use the morning spots on the instruments, which are
Yeah,
typically less more available. So it's much easier to get your.
and less problematic
Oh yeah, the staff
because they're
just
not
finished running QC and you're good to go.
it hasn't been blocked by the person before you running stuff that's been sitting around for 3 days without filtering it.
Right, right.
Yeah. I mean, that was why I started doing it more than the sensitivity was that was just like I was doing these complex animal studies and there were like challenges to assess the function and the disease and then isolation of cells from all these tissues. And then you have your 4 hour cytokine restimulation and then your fix and then your staining. And suddenly you're looking at being there until midnight and then you've got to run the cells and it's already 2 a.m., right?
Right,
And it's
right,
like, well, what's the difference between me going home at like midnight and running these cells at 7 o'clock in the morning and me running them at 2 in the morning?
right.
So
Yeah,
I'm just gonna go home. It'll be fine. It's in the fridge.
yeah, yeah. Does the do you have so what's the kind of workflow that you or I should say the throughput that you have in terms of number of samples that you run in your facilities? Are they pretty heavy sample heavy experiments, like lots of samples to run, or just a few at a time, or like
Yeah, I mean, a typical run for us is somewhere between 100 and 200
oh
samples.
yeah,
We do large experiments, either human or mouse. Usually we have, I mean, our lab is nice in that we do a lot of collaboration experiments between different people. So everybody has their project, but you chip in and help out with other people's projects so that it all goes much more smoothly and everybody's results are good. But then, you've got 100, 200 samples to get through. So you try to start early in the morning on this. I'm going to run it all in one day.
right. Do you guys have a protocol that's and now I'm this is my bane. I can't remember people's name, but there's a paper from a former Mario Rotor staff member who basically described this like the heavy experiments that they run, and it was basically a multiple day um,
Mm,
protocol. Where do you know what paper I'm
yeah.
talking about? And
Um,
what's the name of this gentleman? I love the
Thomas.
guy. He's now a part of a elite group of people that I love, and I can't remember their name for the life of me.
Yeah, it's I'm now also blanking on it, but he's Thomas and he's in
Yeah,
Switzerland and now he's moving to Norway, I believe.
right,
But yeah, he's got this great protocol for getting everything to be robust over time, full details of the workflow over the week. It's a great paper. Yeah.
is that something that you guys nailed down as well, or working on it, or do you too wide a variety of stuff that you do that you can't just
Yeah, for the mouse work, it's
yeah,
too wide a variety. Like
right,
every sort of project has a slightly different approach. For the human work, we have that now. So I've set that up, and I mean, his paper was very helpful in figuring out some of the details that we needed for that. But we have that. Um, Last year, a lot of the stuff that's been on the blog coming out recently has been sort of stuff more related to human work, and that was because last year I was doing an optimization of a set of panels for a consortium. So we need to set up like all of that stuff to make it robust across different centers and so on.
mm-hmm.
Yeah.
That's a segue into something that I'm not sure you've discussed much in your blog, which is but we started making noise about it just because I don't know we get bored and we just want new stuff to talk about. But like the sample automation and basically using a liquid handling device to stain your cells, is that something that you guys have started looking into?
We haven't. I mean,
No.
I've seen some really neat stuff with that. So I know that there's um, the VIB in Belgium has done some really nice stuff with that, and we have some colleagues down in Leuven, Belgium, also who've got one of these ChUG cell washers, and they're
Mhm.
getting great results with it. Um, yeah, I mean, it sounds really good because it also gets rid of some of the debris, keeps your cells sort of agitated, so you get more interaction for better staining. But we haven't done it.
Right. We started dabbling with it, and over the years, I managed to accumulate a an Integra Assist Plus liquid handling device.
Hmm.
Not sure if you're familiar. And it's not an endorsement, but it kind of works for us, and kind of like it. It's kind of
Nice.
a cheap, not cheap, inexpensive. That's what I want to say.
Yeah,
Robot that just moves stuff around, and that
yeah,
actually happens to move the Integra automated pipette, and they have a wide range of those that will pipe at different volumes, and so you can like stick it in there, and the software will basically take into account what you're actually using, what kind of pipette you're doing, and basically lets you do and we got those for multiplexing assays mostly, but like we're going to try to get our users who are doing complex um, uh, spectral panels to run their
yeah.
master mix, stain their cells, the FMO preparation,
Makes a
remove
ton of sense.
any kind of pipetting errors. People
Yeah,
with HDD will enjoy working with this thing. And then just this month, we managed to get the ChUG Alpha to sit next to it. And so plate washing will be automated. So I'm kind of excited to see how that's going to fly. But it's basically,
yeah.
yeah, we're looking for friends in our community to start using those because I don't have the flow through to or the number of samples to justify it.
Yeah, I think it's got to be either something automated like that or these sort of lyophilized panels
Right.
for this, um, to get the reproducibility with these high parameter panels. It's just too hard.
Yeah. Yeah. The question of how to prepare an FMO comes to mind too now because like I've started programming on the Integra, and then I realized it's still me telling the machine what to do,
Yeah,
which is never good. Like I'd
yeah,
much rather have the machine figuring out, oh, you want to do this. Like I can actually find the best, the best way to pipe it, the FMOs, so that you win
yeah.
sometimes.
FMOs are hard.
Yeah,
They're really useful, but I don't do them nearly as much as I should just because they're hard.
right. Hard to prepare, like technically, or just like when you get the data, you're not sure.
Yeah, technically, I mean, it's easy enough to do one, right? But
Yeah,
then doing multiples is hard.
yeah, yeah. We had a nice PowerPoint of my former co-director did a nice PowerPoint of how to do that, but like
Yeah.
it's still just moving tubes around, and it's just like, yeah, it's still.
It's just so many chances of making a mistake anyway.
While we're on the topic of FMOs, and this is like, but every now and then one of my users will show me an FMO that doesn't seem to work well, and it's basically the spread with or without the marker on your negative population will change, and so with the sample, the spread of the negative is actually different than what you have with your FMO. And so it's like the actual marker has an impact of, and so there's where I'm like, I'm not
Is
sure
in
that's always
it's more
it's more, yeah,
when the marker is present.
yeah, yeah. So I'm never quite sure how to deal with that, and I'm always kind of worried about telling my users, yeah, just go back and stain properly because I'm not, I don't, it's not very polite. But I always kind of figured it was like a titration issue, but
Is
I
there
can.
spread below zero increasing as well as above zero?
Um, Good question. I think it's probably the I never spotted a whenever that happens. I don't spot necessarily a on mix like a spectral correction issue. So I would assume it's basically both side goes up.
Yes, I mean, there's sort of two things that come to mind. One is that, like you're saying, that's a staining issue where they just don't have the antibody titrated quickly, so you're getting more non-specific binding in the presence of the antibody, and that will just usually appear right as more sort of fluff around the positive side of the zero. But I think that so I haven't used the S8 and A all that much, but I have seen this thing where single stain controls from those particular machines have an increased variance below zero in the same unmixed data channel. And I do not understand that at all.
Okay. Yeah. All right. Something to explore.
Yeah.
Yeah. Another point that I noticed on your blog is that there's a huge part of it that's dedicated to analysis and data analysis tricks. And you see, if I understand correctly, you do most of your analysis through R
We
or
do.
yeah,
Um, we do. I mean, so our lab is mostly PhD students at the moment. So like
yeah.
they'll mostly still use FlowJo for basic questions. And then my job is to try to make some tools that they can use. And so I mean, a lot of the stuff it's on the blog is analysis because it's something that I'm learning. So I'm on that journey now, and I'm trying to share things that I've learned. And it's also easy to make figures showing what is going on with that,
Mhm.
which I like. And so I can ask the question, and then I can quickly answer it in the data analysis rather than just having to go do a set of experiments. And so it's easier to produce those kind of content for the blog in a way.
Better flexibility and better.
It's something I can do on my own at home
Right.
without having to go into the lab and use resources as well.
Oh yeah.
Right.
Did you learn R to do your analysis, or was that something that came about some other way?
Yeah, just to do the analysis.
Right.
So we had, when I joined the lab in Belgium, Adrian hired a bioinformatician as well, Carlos Roca, who figured out the autospill thing.
Right.
And so he wrote that in R. And so that was sort of the start of me learning R there and seeing a lot of people use it for single cell RNA sequencing. I knew like, all right, I need to learn how to do this because we're getting into this big data.
Yeah. Any tips and tricks on how to get into R? It's something that I always thought I wanted to do, and then it just never happens. But
So I mean, I started learning just in our studio. There's a program that I installed called Swirl
Mhm.
that just walks you through the steps in R. So it's just a course that it takes you through. That's now probably very dated in terms of how you do it. And to be honest, now I suspect that just using one of the AI tools is really the way to go
Right.
because you can immediately jump into learning the thing that you want to learn. Like, all right, I want to learn how to do an R Shiny app to help users select which cytometer to use in my core based on their experiment. It's like, all right, show me how to do this. And then you can learn from that by looking at the output and testing different parts of it and breaking the pieces down to understand what they do. But it gives you that jump start into learning because otherwise it's really hard to know where to start because there's so much information out there and you don't know where to look. And so if you have the AI to help you point you in the right direction for the thing that you're actually interested in learning, you make quicker progress, right?
Yeah. Yeah.
I find that psychologically very helpful. Um, you still have to put in the effort if you actually want to learn it, like not just use the AI to write the stuff for you and not understand what it's doing, but sort of using it to sort of goad you on into learning and keep you motivated to rather than continuously banging into walls where you don't understand something. That's helpful for me.
Yeah. I'll put it in the show notes. There's an actual R course that's going to be online that will start in February.
Yeah,
And again, I forget the name of who's involved, and it's David, but I forget where he's from and everything. But I will put that, and then it's basically free, and
nice.
yeah, it's kind of a great initiative from
Yeah,
the community.
definitely should start with a formal course to get
Yeah,
some introduction to the theory and what exactly programming is.
yeah, yeah. You mentioned Autospill, and it's something that we discussed in our email exchange before this presentation. I came aware of Autospill around, I think it was the COVID years is when it came out, right?
Yeah,
Or
I think that's when we finally published it. Yeah.
because I saw that during I know I was at home and I saw that and it was like okay, and
It was
only a
basically
couple
the
of
end of conventional flow in a way. It's like
yeah.
we published it and stopped using conventional flow.
uh, it took me 2 years actually to come back to it and realize how awesome it is. I'm pretty sure that on the field people are not completely aware of how great that tool is actually, because it saves you so much time, and it makes your mix, like your conversation, faster, and in many cases, if not as good or even better than what you would do if you were trying to manually gate. And so,
Yeah.
uh, can, we'll just a few words on Autospill, and then we'll switch to AutoSpectral, which is the latest version of this tool. Um, so essentially, Autospill, like what was there a specific problem that you faced whenever you guys designed this way of gating your, or basically looking at your single stand controls?
Yeah. I mean, so I started out doing two color flow on the fact scan,
Right.
right? And there you have to do manual compensation. You take the mouse and you tweak the values until the data looks straight.
Right.
And I think at that time we didn't even have bi-exponential displays on there, so actually we were probably doing it all wrong anyway.
Yeah.
Um, but it didn't matter because you were doing two colors really. Um, and so that was how I learned. And I had done manual compensation starting from two colors and then just kept working up as we got more and more colors involved. And every time I went to use one of the automated tools for calculating the compensation off the controls, it was a mess for me, at least. Um, and it was always a mess to try to use beads for me as well. Like I never understood why people were using compensation beads because they never worked for me.
Mm.
And they've gotten better and I've learned a bit more about how to use them. They're better now, but they're still not quite right. And so I was trying to build these symphony panels for sort of 23 to 28 colors and doing manual compensation because I couldn't get the compensation in Diva to work or in FlowJo. And that was taking a while. I think the first time I did it, it took me a day and a half to calculate the compensation matrix by hand because it was sort of this iterative process where you go through each fluorophore, adjust against, n by n plots, and then go back and redo the loop again because every time you make a change somewhere, it can percolate through, right? Um, and so I explained to Carlos, the mathematician, bioinformatician, what I was doing, and he realized how incredibly stupid it was and how a computer can do this so much better because basically I was implementing a very basic, like a for loop on the machine. Um, and so he took what I was doing manually and wrote it down in code and made it work fast. And that was what we did for Autospill. And it was better. And I am thankful that I do not have to manually calculate compensation matrices anymore.
Yeah, that's very nice. The so the limitations there is that you, within your controls, you need your positive, negative, everything. But like, is there any since we're doing a linear regression, um, do you actually need to have a unstained, like if you were just looking at like a bright and dim, that would be actually probably better.
Yeah.
Yeah.
Yeah, so I think that's in the paper. I'm not sure that you
Yeah.
don't need that. So you no longer need positive, negative. What you do, so the same sort of rules, Mario Rotor's rules for compensation apply. Like you need bright cells.
Mhm.
And I think that when we talk about bright cells, what you need is actually the full dynamic range. So having a cold negative and having cells that are as bright as what's in your sample is probably better than having something that's, well, it is better than having something that's like between 10 to the 5 and 10 to the 6 only.
Right.
And running regression on that's not going to get you all the information.
Right.
But Autospill will do better with that information than a positive negative because it is calculating more information than just one median versus another median.
Right. Right. And I'm kind of an idiot, but I thought that, so I it's been picked up by a number of platform for analysis. So Flojo has Autospill implemented. Other groups like the Miltony Tido, whenever you do your compensation on the sorter, is going to use Autospill as well. And groups like Florida.io, this
Yeah,
free. So it's kind of it
nice.
got picked up quite a bit. Um, but quite, I don't know if what Flojo did, but like Flojo has a bunch of check marks whenever you do your compensation or your own mixing, and you can check traditional or autospill, but you can also check spectral. And I kind of assume you could do autospill with spectral data in Flojo, and then, but then I saw you come in with
Yeah.
autospectral, and I'm like, oh, was I wrong there? Was that
No, you're not wrong. Um, so when I started trying to adapt it, I first thing I did was go to FlowJo and try to use their auto spill spectral.
right?
Um, I have a good PC. I got it to work maybe one time in 10 without it crashing.
Hmm.
Um, and it wasn't producing good results,
Oh, right.
for me at least, with
Yeah.
any of the data that I put into it. And so I did not understand why, and that was made me want to get into the innards of understanding why, what's going on. And I still don't know what's going on in FlowJo because the basic approach of autospill in autospectral does a decent job, um, and doesn't run into the problems that I see in the FlowJo implementation.
Yeah.
But uh, yeah.
Can you go? It's like what were the tweaks that you needed to implement for to switch from autospill to autospectral?
So some of it's just trying to make it a bit faster
Mhm.
because there's more colors and more data and more channels. And so if you run a robust linear regression in, say, 20 parameters, it takes, say 20 seconds or whatever. If you do that on the Sony where you have 190, it's going to take now 190 seconds.
Right.
And it's going to be proportional to data size. And we know from Mario that you basically need the bright cells and the dim cells. And so one of the quick steps is just, well, let's take just the brightest 1% and some matching negative cells and run the regression between those and cut out the middle, which is much more similar to the positive negative approach. But we're still getting the benefit of the robust linear regression, which tells us, well, some of this information is less accurate than the others. And so some of that information, particularly with cell controls, is autofluorescent events that are really not your fluorophore signal.
Hmm.
And the robust linear regression says, well, let's keep to the ones that look like they're close together along the expected dimension. And if stuff that kind of looks like it's floating away, like macrophages or monocytes, let's pay less attention to that. And that works better than just taking all of it in a median. So there's that. And then I implemented some other steps that we often have to do manually when you're working with cell controls on spectral because of where the autofluorescent cells again intrude. So depending on where your fluorophore is in the spectrum, now you have so many more choices than you did with conventional. You can get a lot more fluorophores being interfered with by, say, macrophages in your splenocytes or monocytes in your PBMCs. And you can do subsequent gating to focus on, say, your T cell marker that's near your macrophage at autofluorescence. And so there's one step in autospectral that deals with cleaning up the controls so that we can exclude those cells that really aren't the fluorophore signal to prevent them from contaminating our definition of what the fluorophore looks like. And so that's the parts just focusing on how do we get the best information out of the single color controls. And so the second half of autospectral is saying, well, what do we do with the information that we get out of the single color controls? How do we apply that to the fully stained sample? And I I think that the insight there is that we don't have one signal in autofluorescence. Like we know that, right? We have multiple autofluorescences present. That's also true for the fluorophore signals. There's a distribution of information. If we just take that median signature, sort of the middle, we're going to be accurate. We're going to line things up correctly, but we're not going to be very precise. We're going to have that distribution
Right.
of spreading. That sort of it's part of it, we can think of it as spillover spreading. I don't know if it's exactly that, but it manifests as spillover spreading because you have a distribution of possible outcomes in terms of flow formation, more red, more blue. And that, depending on what it's similar to, may give you some spreading. If you sort of take into account that distribution of what's possible from the single color control, which is actually a very, very narrow distribution, we can then look at the single color, the fully stained sample, and for any given cell, sort of figure out where it is on that distribution, which means that we can use, if it's more skewed to the blue, we can bring it back towards the zero as opposed to being sort of skewed blue. Does that make sense?
Yeah, yeah, yeah. Thanks. I know a couple of people on Discord that mentioned starting dabbling with Autospectral. Do you have any tips on how to deal with the algorithm and any warnings or any tips on how to make it work as best as possible?
Yeah.
Yeah.
So, I put a bunch of hopefully helpful articles on the blog and also on
Mhm.
GitHub with practical examples of how to work through it. Um, they're probably not great because to me, because I've designed it, it's fairly obvious on how to use it.
Right.
So I'm trying to make it clear, but I've probably missed some stuff. It's also the first time I've done something like this. Um, the first time I've written an R package. It's not great. I'm aware of that and I'm trying to fix it. It's also quite slow, particularly the part where we're trying to figure out where the cell is on the distribution.
Mhm.
Um, I figured out a trick for that I'll be implementing in the next version that should make it hopefully quite a bit faster. But I appreciate that it's probably not great code because that's really not my forte.
Right.
But yeah, if you got questions on how to do something specific that's not covered in the articles, yeah, reach out and tell me what you're struggling with about it, and I'll try to put something together on it. It might take a while,
Yeah.
but I want it to be useful. I want it to be helpful, and I appreciate that it's not probably great. I'm kind of hoping that if we get it published and it's useful and correct, that somebody who's better at coding can make it work a bit better.
Yeah. Yeah. It's my approach to podcasting. I'm hoping somebody will look at what I do and decide, oh, this is fun, but I can do it better, and basically take my spot and
Yeah,
do it more often.
that's all we can hope for,
Generally,
right? Is
yeah,
like this is what I can do.
yeah,
Can you do
yeah, yeah,
better? Fantastic.
yeah, yeah, yeah. I wanted to use the last stretch of our time together to discuss basically the field of training. I kind of see your blog as one of over the last few years. There's a few things that I think are going to be have been great for the field. One is Discord, but the other one, your blog has been super helpful to so many people. So I kind of wanted to pick your brain about where you see things. Do you see any gaps in the training that's offered to the people running into getting started with flow cytometry? I think I was mentioning to you that my it took me forever to understand how Autospill can be used properly and how useful it would going to be. And I talk to my colleagues every now and then, and a lot of them are not still familiar with Autospill and what is how to use it. So I get a feeling that the training on the field has been if it's done by core facility, we're going to go back to the stuff that we already know and how to compensate and all the stuff that the treaded path, or otherwise we go to manufacturers training, and there it's like SciTech is a good example of a group that's really well involved and provides useful information.
Yeah, they got some
And
great
then
stuff online.
Right, right. But then you look at Sony, and Sony is I think they're changing their minds about the way they've been going at it, because they provide great tools on their platform. The ID7000 has great unmixing tools, but like very few people understand how it works or why, and so it's kind of like there's quite a bit of work that I feel should be done, and I'm not sure how you see it.
Yeah, I mean, for me, I think the big one is trying to understand how you're going to use the data at the end and designing your experiment to allow you to do that in the analysis. Like thinking through the full process, like not just like I want to stain for Tibet, but like what am I going to be doing in terms of analysis? Is it going to be gating? Is it going to be pre-processing? Is there going to be some sort of harmonization of data sets from multiple centers? Is it going to be this instrument? What is my gating strategy? What is my clustering strategy? If you understand those things going in, you can design your assay to better equip you to use the analysis tools or to avoid problems that will prevent you from using those tools effectively at the end. Um, I think there's some really good work going on, particularly from uh, particularly people like Sophie Van Gassen are doing really nice stuff on showing how the tools work in a sort of broad overview way, so you can think about them and incorporate them into your workflow. And just having sort of that level, even if it's cursory understanding of what is available to you and what you're going to use before you do the flow cytometry experiment is super helpful, I think, in getting good results out and not ending up in a situation where 3 years down the line you've collected 2,000 FCS files and don't know what to do with it. And you
Right,
have to start thinking for the next 3 years about now what do we do?
right, right. You mentioned a couple of times during this our discussion together, like the choice of instrument, and what do you mean by that? Like I kind of always tell my users like the instruments in our facilities are pretty much like sturdy enough that we can do fairly complex stuff, whatever. So it's not. When I started, we were basically having fax scans all the way to LSR2s, and now we needed to figure out, okay, how many markers you need. Nowadays, that's not really the issue. So what when you think about the choice of instrument, what are the main criteria that are going to drive your decision?
Yeah, I mean, I'm probably still thinking in terms of the past here, where
Right.
we have both spectral and conventional instruments available, and sometimes just doing something simple with two colors on a conventional instrument is what you need.
Yeah.
Um, yeah, I mean, to be honest, mostly just use the Auroras that we have in our facility. That's all the students get trained on now. We have two different types of sorters, so um, that's probably the one where they need to think more about which instrument they're going to use and design around the capabilities of that instrument. Um, yeah.
Yeah. No, so yeah, that sounds about right. The in our facility we have a range of platforms. We were BD heavy just because BD was dominant, but like over the last few years, we started branching out, and so we have Agilent platforms, the Cytec Aurora,
Mhm.
and others, and now we're just thinking in terms of okay, what are the features? What are you actually trying to do? And what are the features on the instrument that will benefit you. So the attune goes fast. So if you're going to screen a whole lot of samples, that's what you want to use because like there's no fun. It's not fun to like run tons of samples on a machine that's like runs everything at turtles pace.
Yeah.
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 We had a podcast with Florian Mayer and Ree Gardner on bad on mixing results and
Uh,
the debate about do you mess around with the results or
oh,
do you just let it go?
yeah.
Do you have an opinion on that? Mine got since the podcast got tweaked. We had a Sony demo of the ID 7000, and they sent me. I think they listened to what I said for some reason, and they realized I was wrong about everything, and they
Oh,
sent like really heavy caliber people to the facility to explain to me how Sony works, and I funny like I did a we had a GLIFCA meeting in Chicago. GLIFCA is the Great Lake Association of Imaging and Flow Cytometry, and it's like a yearly meeting. And I was driving a discussion in there, uh, everybody had canceled, so I was like left to like fill a gap, and I made a discussion about that topic. Like, do you mess around with your unmixing results? And at that point, I didn't understand quite well how Sony uh, resolve some of the issues that you might have at the outset of your unmixing process, and you still have issues, but like you can correct it somehow. And I was I was making the claim that I was using a metaphor for it's the trolley
Mm.
problem that I put in. So if you have like one person on one track and five on the other, or one person on your track and five person on your track and then just one on the other, do you pull the lever to like go murder
Yeah.
that one over there? And but then if you turn the situation around and there's only five, like only one person on your track and then five on your, you don't change the lever because like,
Yeah.
where does that make sense? Oh no, if the one to one is the, I'm messing up my metaphor completely.
Okay.
If one on each track, you don't change the lever because like
It
at
doesn't
that
help.
point you're taking responsibility for
Yeah.
and so that was my position on messing around with your mixing, applying compensation, or using something like the tools from Sony. It gets you to the same results. You still
Yeah.
mess around with the data. In one way, you just like manually did it, and the other way, you just let the software do something. Um, but
It
it turns
achieves the
out,
same result. Yeah.
but it turns out Sony, and you've tested the Sony platform. Do you have one in your facility, or you just did a demo, or
We don't. There's one at Baberham where I was before, so I've
right?
used that one. And we have one that our collaborators operate down in Leuven, Belgium. So I use
Right.
that one sometimes. Yeah.
So the way Sony does it is basically it change the signature. Like instead of applying
Yeah.
compensation like you would do on other softwares, it changed the signature of your
Yeah.
single
Which
stage control
recalculates the
and
unmixing
recalculates.
matrix.
Yeah. So
Yeah.
that's the main difference. So it's now you can track the changes you've made to your data. So in a way, that's somewhat
It
better.
is fundamentally the same thing. You can
Right. Sure. But you can
back calculate the
track it. Is
compensation
my
changes to
yeah.
correct the spectrum
Right.
and achieve basically the same thing.
Right. So would you do it or not so much? You just don't touch it, or you where do you
I
sit on that?
try to avoid it.
Right.
So I haven't done it in a few years now, I think. For me, that indicate if you see that there's something that looks like an unmixing issue, you need to go back and first check what's going on with the controls. Like that's the first thing that you should do is understand what's happening with the single color controls that's causing the problem. And if there's cells, oftentimes there's a lot that you can do to improve it, which is, the kind of improvements that AutoSpectral does automatically. If you're working with compensation beads, usually it's because one of the beads doesn't actually reflect what's going on in your sample.
Right.
And so there, I try to tell people like, look at this. If you see something's funky, have me check it in our lab and rerun the control for that if we can't fix it, like, and do it soon. Because the sooner you can rerun the control on that machine, the more likely it is that you'll capture something correct to what you were doing, and then re-unmix using the new control. And most of the time that fixes most of the issues. And the only time where I personally feel comfortable applying some sort of correction in the unmixed space with compensation is something like a PE tandem breakdown,
Right.
where it is specifically between the tandem and PE, and I know exactly what's happening.
Right.
And I can understand like chemically what has caused the issue, and it is because there's more tandem breakdown in the sample than the control. And that is very difficult to replicate with the control. Otherwise, I just try to get better controls or change how we're processing the controls.
Yeah. Autospectral should help tremendously in providing better on mixing, right? Regardless
It
of the
should.
algorithm you're going to be using.
Yeah. I mean, that's the aim.
Yeah.
So it doesn't fix stuff where you're using
Bad.
beads, right?
Right.
It
Yeah.
doesn't fix stuff where you've added like two colors to control.
Right.
Doesn't fix it when you haven't added the colors of the control. I mean, these are actually kind of common problems, right? And that's the problem is a lot of time is that the controls are just not great. And it doesn't fix the problem if the control's not bright enough, or if you've got different tandem breakdown. It can help a bit with all of these things. It can bring it a little closer to true, but it doesn't necessarily fix it all. Maybe we'll get there. But a lot of it's, for me, a lot of it is like if you see an unmixing error, it means that a lot of the time it means that you've done something that's not quite right between the controls and the sample.
Right. Last question for you, just to open up a bit, but like is there anything that you're looking forward in newer technologies, new development in the field that you're excited about, that you see coming, that is going to be helping you in your work?
I mean, no, to be honest, the technology is moving so fast that I
Right.
expect that the next innovation that's going to change things, I have no idea about what's coming. And I'm
Right.
super excited for that.
Yeah. I was hoping, like I was expecting an AI answer, but actually, I'm a Luddite on AI. I'm not sure.
I mean, it's been really useful for me
Yeah.
to get to be able to do things really quickly and learn more. And like I said, not get frustrated with the coding because it's a slow process.
Mhm.
Um, but that's about all I use it for is situations where I can verify the results.
Right. Well, Oliver, it's been a pleasure talking to you. Thanks so much for your time,
Thanks
and
for having me.
pleasure. Like for a listener, thanks for hanging out. Like and do the things on the thing, and we'll talk to you later. Cheers.